Mutahara. So we already have 2 questions in the chat box. So if anyone want to ask more questions, please do so. So the first question is from Abdullah Al-Asif from UPM Campus Bintulu. So his question is that he is working in existing Malaysian law. So his question is on the law. So do you think that the existing law are supportive of protecting the seagrass ecosystem in Malaysia? Because he thinks that the area coverage of seagrass in Malaysia is very small compared to the other countries in Asian countries. So what do you think, Prof?
Okay, thank you for the questions. We have to look here. For example, we have marine protected area in area Pulau Redang and also in Sabah, in Sarawak. In terms of other area in Malaysia, we already have the protected area, including the marine environments, that one. But if we want to protect, for example, I give you an example in the Tanjung Adang Laut Show, or we are called Sungai Pulau Esri. We have to think the local people because of the traditional activity, fishing activity here. In that particular area is very important for them. So that's why the important thing is we have to give them awareness. What's so important about that one, and then how to preserve that one. But nowadays the problem is now in Malaysia, people always not understanding about the seagrass itself. They always think, okay, the seaweed, the animal, but they don't understand. Before the animal come in, the most important thing, we have to awareness, we have that one. So that's why when people already knows about that one, then the protected area, we cannot allow the laws to cover every area, but we have to the certain area nowadays. We have cover in marine park. That's all from me.
Okay, thank you, Prof. So he also has another question from Abdullah too. So he mentioned that the German government recently launched a 1 billion USD fund to protect biodiversity all over the world, along with German territories and to the countries that have bilateral diplomatic relation with Germany. So he also attached the word links in the chat box. So his question is that, is there any such fund in Malaysia that allowed Malaysia to apply to protect the extensive biodiversity and the restoration of this biodiversity?
Okay, just for your information, actually in Malaysia, we have studied. That's why I started 1989 when I graduate. I'm study about the seagrass. Take with me in 1995. We study about the distribution. We study about the ecology. We also study about the genetic. We also study the transplanting. So together with part, so that's why we have also APN project, Asian Pacific Network project, Seagrass Watch, and other thing. So actually we have many what we call sponsor grant, but we must understand they must always want to give that one, but we have to have the evidence. And in Malaysia we have CTI project, Coral Triangle incentive. So we have that one. So also cover not only per se seagrass, but we have to cover everything there. So we have that one. And also we also have the committee member about the everything this in terms of seagrass, seaweed, and also coral reef, and include mangrove.
Okay. All right. Thank you, Prof. So I think there's another question from Dr. Nur Salwa from UMT. So her question is that with the conservation of seagrass in Merambong, did our dugong come back or play at this area, Prof? Is there any sighting recently?
Okay, just for your information, in the Sungai Pulau Esri, not UPM group study about the dugong. So other people in Johor, in other agencies study about the dugong, and then yes, they also discover the dugong come in when the seagrass is very healthy. So they already published. For example, you can see in the website of the Safe Seagrass. I'm not remember what is the person. Might be Leena knows. It's not Dr. Serena, other person. And then I'm also joined the activity, how to protect the seagrass, especially for the Wong, when they have a seminar and conference on holistic of the seagrass area.
Okay. All right. So I think they can look through it later. Maybe from the internet there will be a lot of information.
Yes.
So we move on to the next question. Is from Susan Putin. She said, "Great presentation, Prof. Who do you work with in Sabah? Do you work with UMS?"
I work with UMS, Universiti Malaysia Sabah, and also Sabah Park.
Sabah Park.
Yes.
Okay. All right. So is there any more questions before we move on? So if anyone wants to speak up, you can. Yeah, sure. Please go ahead.
Okay. My name is Lin Lin Hashim. Assalamualaikum, Prof.
Waalaikumsalam.
Okay. Thank you for your excellent talk. In your opinion, do you think seagrass in Malaysia can adapt to climate change?
Okay. Just now I mentioned, I'm really monitor, thorough checking the seagrass within 5 to 6 years since 2014 until now. Monthly, I visit that particular area. Yes, seagrass can adapt to these environments, to the climate change, or in Malaysia, we call the temperature, the sedimentation. People always mention if the sedimentation is high, the seagrass cannot survive. But in Merambong Shoal, the seagrass, the total suspended solid until 200 plus, 100 plus. But the seagrass still healthy. Why? Because of still have a current. And then in terms of tropical monsoon and so on, is very helpful for the seagrass. And of course, except for Telaga Simpul, for example, because of really we developed that one, that one is gone. In terms of climate change, yes, we can.
All right, thank you.
Okay, thank you, Prof. One more question from UMT, Miss Afifa, Dr. Afifa, I think. She's working on Bivalve in Setio since 2012, and she see the changes on what you mentioned just now on the seagrass loss, and it affects the mollusks, especially cockles, which seems no longer or hardly find there. Is there any plan to recover the seagrass in that area?
Okay, that's why we've now since 5 years we cover area in south area. Why? Because we have to divide. People also we have expertise in Terengganu. That's why they need to focus there. For your information, of course, when the seagrass is reduced or not healthy, the mollusk, the organism also affects. So if you want to study, make sure we have to know the in situ parameter about in that particular area, culture in the laboratory first, then you can study this one. The important thing is the initiative from the individual, because we not enough time to go Johor and also that one. But for this year and next year, we will focus in other area, not only in south of Johor.
Okay, thank you, Prof. One more question from UPM Campus Bintulu is from Dr. Tan Tohi. So his question is, have you tried seagrass planting in Bintulu area?
Oh, for your information, in Bintulu area, I'm not tried the transplanting. Why? But I'm cultured in the indoor and outdoor laboratory systems. Because of the seagrass in Bintulu, usually in the deep water, for example, Halophila decipiens in the area, and then another one is Bintulu also is related with the crocodiles. So it's not, but in the Lawas, Sarawak, we not conduct the transplanting, but we monitor the seagrass together with the forestry departments. Okay.
Okay, thank you, Prof. So I agree with you regarding the crocodile problem there. It's quite difficult to do transplanting there with the crocodile roaming around. All right, so another question from Nadia. Does the disappearance of seagrass in certain area cause algae bloom?
Okay, some cause of the algal bloom. Some, for example, when the nutrient is very, very high, so the species, the popular species, is what species? Ulva reticulata come in. So that's why the seagrass covered by the ulva, but don't worry, because as ulva species actually is a floating plants. So when the what we call when the current come in tide table, so the seaweeds become removed, that one. And then of course seagrass can stay in that particular area. We must remember they produce by regenerate by their rhizomes in the soil. That's one is very important. When we can culture by using only rhizome without leaf or rhizome with leaf. That's all.
Okay. Okay. I think that's all questions I can get from the chat box. If there's any more question, please speak up now, or we are going to move on to the next speaker. But anyway, thank you very much, Prof. Putra, for sharing your knowledge and your experience, many years experience working with the Malaysian seagrass. So thank you all. And now we want to move on. But before we move on to the next speaker, I want to do a small announcement. So we are collecting names for the e-certificate. So if you would like to have an e-certificate, please fill up your particular in the attached form in the chat box. So you can find it in the chat box. There is shared by Nur Shahira, our staff. So there's a link that you can click on for the attendance form. Please just fill up that. So we will try to issue a e-certificate for everyone who attend here. Okay, thank you very much, Prof.
Welcome.
All right, so now we are moving on to the next speaker. So Prof, have you closed your presentation?
I'm not close yet. Is it?
Yeah, wait. Maybe it's my problem here.
Or might be I have to leave first. I don't know.
No, it's okay. I think just close is fine.
Okay. I don't know how. Because I have problem with internet now. Oh my God.
It's okay. I think Dr.
Okay.
Firdaus?
Okay, I'm stop sharing now.
All right, great.
Thank you. Sorry.
Thank you, Prof.
Okay, welcome.
All right, next we have Professor Julia Sidward. Is Julia? Yes, she's here. Hi. All right, so we'd like to invite Professor Julia Sidward for as the next speaker. So she is the head of malacology at Senckenberg Research Institute and Museum in Frankfurt, Germany. We are pleased to have her today. And she works on the evolution of diversity in mollusks, and she has authored a book titled What Species Mean? A User Guide to the Units of Biodiversity in year 2018. She has named 13 mollusk species, including 2 fossil species, and with more than 100 publications. And her research interests in genetic, morphology, anatomy, neurobiology, and physiology of marine mollusks, including both tropical and temperate species, coastal and deep sea species, has contributed greatly, significantly to the understanding of mollusk species evolution and diversity. So without further ado, please welcome Professor Dr. Julia Sidward with her talk titled Advances in Understanding the Impacts of Global Change on Mollusk Biodiversity. Yes, over to you, Julia.
Okay, thank you very much. I'm very happy to be here. I'll try to share my slides now. Okay, does that look all right to you, Leena?
Yes, great.
Fantastic. So thank you very much for having me. I'm excited to be here virtually, although I'm sad not to be able to be in Malaysia in person. I'm really looking forward to the end of this pandemic and a time when we can be together in person. So I wanted to talk to you today about the diversity of mollusks in Malaysia and the importance of understanding taxonomic biodiversity in a broad sense for understanding the importance of ecosystems in Malaysia, in coastal areas in Malaysia, and also for the role that Malaysian biodiversity plays in global biodiversity. So for some context, I think it's important to think about the difference between the land and the oceans. So in fact, there are many more species on land than there are in the oceans, but that's because there are most of the species, the animal species, I should say, the animal species on Earth are insects. So we know that insects are the biggest group of animals. There are millions and millions of insect species. But in fact, the diversity of life that we have on land is much lower than the diversity in the oceans. So if we think about just the different groups of animals, the different higher level taxa, the classes and the phyla, there's much more diversity in the oceans than there is on land. And yet, because we are terrestrial animals as humans, we walk around on land most of the time. And so we think more about mammals and birds and insects because these are the species that we see. And we sort of assume very often that, you know, the life in the oceans is more homogeneous. It's more the same, you know, in different places, and we can't see the subtle differences that we can see when we move between different habitats on land. And this is a problem because it means that we put our ocean ecosystems at risk because we don't understand immediately when they are unique and when they are important. And it also is an important scientific risk because we need to understand as much as possible about diversity in the broadest sense to really be able to understand life on Earth. And this is what I mean about the diversity of land animals versus marine animals. All of these groups that are showing up in green, they have representatives on land. Okay, so there's us, and there's some land mollusks, and there's earthworms, and and other groups such as arthropods with the insects. But all of these groups also have marine representatives. And the ones that are not highlighted, the majority of groups, are only found in the marine realms. So there is actually much more diversity of different animal forms, different life forms that we find in the oceans. And in particular, I work on mollusks, and mollusks are the most diverse of the most diverse. So mollusks are a phylum, an animal phylum, that is the second largest in terms of species after the arthropods. But again, the arthropods are bigger because they have insects on land. And mollusks are incredible because they have far more morphological diversity. So all of these different forms are different kinds of mollusks, including bivalves, gastropods, cephalopods, but also these several other forms such as aplacophorine mollusks, scaphopods, the chitons, the polyplacophora, monoplacophorines. So there's more than 100,000 living species of mollusks in these 8 separate taxonomic classes. And these reveal a long evolutionary history with lots of diversification, reversals, convergence, and many novel forms, many novel adaptations of how these individual species have accommodated their environment, in some cases in quite extreme forms. And I'll show you a couple of examples of that from work that I've done in Malaysia as we go along. So even the more familiar groups of mollusks actually contain an incredible amount of diversity. So the gastropods have a fossil record from the early Cambrian to now, to the recent, so the entire span of the evolution of animal life on Earth. And they have these not just normal snails, like the one in the middle, but actually this incredible diversity of forms. This crazy coiling thing in the upper right, this is a tiny land snail from limestone karsts in the Bantu caves that's endemic to Malaysia in these tiny, tiny spaces. And it's unlike anything else found on Earth. It coils like a crazy thing. And then there's many other different forms of gastropods, from the cowries that have these hard shells adapted to avoid crushing predators, to the scaly-foot snail at the top, which is a strange snail endemic to hydrothermal vents, or Xenophora at the bottom left, which is a snail that actually makes its shell by picking up the shells of other snails so that it can camouflage against the reef, or nudibranchs, like the one at the bottom. This is a dorid nudibranch, but the X-ray image at the far right is the skeleton inside the nudibranch. They actually have a complex skeleton made out of spicules. And then there are gastropods that have actually given up on looking like a snail completely. So this is a strange animal that some of you may have seen before. Dr. Leena and I have worked on these animals for several years now. This is Berthollinia. This is a bivalved gastropod. So it's a gastropod, it is a snail, but it grows a shell like a clam. And I hope you can see it crawling around, and as it moves around, you can see that it has two halves of the shell that literally open and close like a clam. It's not unique to Malaysia, but this is a group that lives around in the tropics around the world, but they're very rare, and Malaysia has at least one species that's found mainly in peninsular Malaysia, and perhaps several more in Malaysian Borneo. So we have worked on this animal and the importance of this strange bivalved form to understand the way that these body forms can adapt to withstand environments in this case in seaweed, not in seagrass, but these very turbid and turbulent environments, and what special features the bivalve gastropod has that allow it to cling on to the seaweed, and to what are the drivers for inspiring these strange and awesome body forms. So bivalves, real bivalves, don't have a head, they don't crawl around, they're not as cute, but they also have quite an incredible diversity of forms. So we think, oh, clams and mussels, you know, I know about these, they're good to eat, but if we stop and think about these things, even though they're familiar, they're still incredibly diverse. Scallops, like the one at the top right, have eyes that are unique among all animals that have a two-part lens system. There are some clams that are actually carnivorous. Mussels that we find on rocky shores make the byssal threads are one of the most strong natural products that have ever been found. And giant clams like the one at the bottom, these are actually photosynthetic. They host symbiotic dinoflagellates in a similar symbiosis to corals, so they're actually getting part of their energy from photosynthesis. And then there's others at the bottom, like the one at the bottom left, pholas, which are actually able to bore into rock. They have an incredible acid-secreting gland that allows them to bore straight into rock and live inside rocks. And then there are bivalves that have given up on shells altogether and live like worms, like the shipworms that bore into wood. So I'll give you a couple examples of bivalves and the work that we're doing now on some extraordinary bivalves from Malaysia. But to put this diversity in context, we do understand a lot about the evolution of bivalves and how living bivalves form into 6 separate major groups. So this is work that we've been doing on the genomes of Malaysian bivalves with a couple of my colleagues, Carola Greve, who's the lab manager here at the Genome Center in Senckenberg, and also Vanessa Gonzalez, who works at the Global Genome Initiative in the Smithsonian in Washington, D.C. So we looked at some examples of 2 of these groups, 2 of the weirdest of the weird, the Anomalodesmata and the Inequidonta. So we were lucky enough to collect examples from these 2 groups a couple of years ago, and we've been sequencing the genomes of these 2 extraordinary species. So the one at the bottom is Scintilla philippinensis, and this is a funny little clam, which colleagues in Japan have actually shown that their strange brown tentacles that you can see waving around, they actually use these to mimic nudibranchs, or maybe to mimic a crab, to hopefully look less like a nice tasty bivalve. So these are adaptations of the soft parts around a more or less normal looking clam-shaped body. And then the one on the right is one of the most extraordinary bivalves of all. This is Verpapinus, it's a clavigellid, and these are clams. You can see on the top of the big white shell, there are 2 tiny nubs, and this is all that remains of the bivalve form in this bivalve, but this is honestly a clam. So inside this tube, there is a body that looks anatomically like a bivalve. And they live in mud. You can see from the little inset picture, they actually live in seagrass shoals, and they make their tube in the mud, and then they can breathe through the top of the tube, just this tiny piece sticking out. And so these are an incredibly rare species, and in fact, they're possibly, as from this quote from Brian Morton in 2007, they're arguably, although amazing, among the rarest of living animals. And they've been recorded since the 1800s, possibly even earlier, from the Malay-Thai Peninsula, but actually in the 1980s and 1990s, they were presumed to be extinct in Singapore. And it was only works by some colleagues in 2011 in Singapore, where they went out very deliberately to try to determine whether this species was still present around Singapore. Although we know that there's a nice, healthy living population in Marabang Shoal, which is, of course, the same place. So we know that they're there and they're healthy. And now, for the first time, we have a genome for this species, which isn't published yet, but I'll show you the basics. I won't go into details to explain this, but what we're seeing here is a very, very nice genome. We're very pleased with these results. This is unusual for mollusks to have such nice, high-quality genomic sequencing for an animal that was collected in the wild, not grown from a culture. And this is, in fact, the first time a genome has ever been sequenced for this subclass. So it fills in a very important piece in the bivalve tree of life to understand the diversity of bivalves in general, and especially that will allow us to understand these adaptations to this strange body form that no longer looks anything like a clam. And there's a broader lesson here too about these strange species, which is that there's so many species in Malaysia that the species far outnumber the taxonomists that we have to study them. And many of them are undescribed. Many species in tropical Southeast Asia have never been named by science. And some estimates from, for example, from Maria in the tropical Pacific, it may be, I think this is very likely, around 30% of the species that we actually could encounter in Malaysia in the marine realm have never been described. And there are many reasons for this. There's cultural reasons and there's scientific reasons. And principally because the history of taxonomy is Eurocentric. It's white people like me who show up and go, I already know what that is, and don't look in enough detail at what is the local ecosystem, what is the local diversity to really understand when species are unique. And there are issues with the names of species and species groups that actually are mainly from the Atlantic, but those names are incorrectly applied across the Indo-West Pacific, which makes it more difficult to find out when a species is new when working in tropical Southeast Asia. So we know that this is a problem. So for example, the figure in the bottom left and the bottom right, this is comparing the fauna of mollusks from Northwest Europe, so around Britain and Ireland, to the fauna around Malaysia. So we have this figure with the sort of the color ramp from like pale to darker red, showing the relative diversity across the world. And you can see always in Malaysia, it's bright red, the most diversity of anywhere in the world. But if we look actually at the number of records in museum collections and in published checklists, it tells a different story. So this little bar graph in the bottom right, there's actually more genera of mollusks, the genus level groups, that have been recorded in Europe than there have been in Malaysia for mollusks. And this is simply not correct. We know that there are far more species and far more genera represented in Malaysia, but they're not captured in the literature. So we know that they must be there, but we don't always know what they are. So there's a big discovery gap. There's a big gap of animals that have yet to actually be recorded from the local area to really represent the diversity. So this is a problem for understanding diversity and for protecting local ecosystems. And it also is important to reflect on why this is. So I said that there's social or cultural or historical reasons, but there's also real scientific reasons. And the biggest one is vicariance. So this refers to the fact that many islands, the geological patterns, water currents of the oceans, these all create boundaries that shape species distributions. Prof. Harah mentioned this, talking about the Malay Peninsula acting as a barrier for Halophila, that creates two different populations of Halophila. And just like in the seagrass, we can see that the past literature often assumed that many species were widespread across the whole Indo-West Pacific. And new research using molecular methods constantly finds that there are many cryptic species groups. So we think this is one species, and then we realize, no, actually it's 3 species or more species. And this is a legacy of Eurocentric taxonomy, but it's also a legacy of tectonic drift. It's part of the geology. So the lower image now of the globe shows the Earth from 50 million years ago, which is about the time that many of these marine species, these mollusk species that I work on, would have already been established. These are, in many cases, ancient species. And so you can see the Indian subcontinent on the left, Malaysia in the middle, and the Philippines off on the right. These are historically, 50 million years ago, these are completely unconnected, totally different landmasses where there's no possible way for species to transit from one to the other. There's deep ocean in between them. Now tectonic drift has moved these different parts of our geography closer together, but the species in many cases are still staying where they originated in these separate places. All of this adds up to mean that species in Malaysia are actually far more unique and far more diverse than was appreciated in the historical literature. And here's another lovely example of that, which is a project that Leena and Yusuf Eza and I have just been working on that's in press now. We're excited to see this paper come out very soon, which is the Muar oyster, found in the estuary of the Muar River, just at the northern part of the state of Johor. So the oyster is on the right, but what's extraordinary about this oyster, it looks like an oyster. So to Leena, it looks very different. Even to me, it kind of looks like an oyster. But what's amazing is that the local fishermen have always been able to identify the species, and we have found historical records in the accounts from the 18th century that this oyster was fished for at least 160 years, and the local community have recognized it as a separate species and as a better species with a better flavor for more than 160 years, and yet it was never named. It never had a scientific taxonomic name until 2021, and the paper isn't even out yet. And now this is a species that lives only in this one tiny river estuary. There was a second population that's mentioned in some historical accounts, but that we cannot find, or it doesn't seem to live there anymore. And we have a lot of thoughts about, where else it might be. If perhaps it's over in Sumatra and Indonesia. But there are no other records of this species except for this one tiny estuary. And Malaysia is absolutely full of stories like this, some that have been told and many that have not been told yet. So I want to mention the role of museums and research collections in trying to get a handle on all of this species diversity. So museums, research museums, are physical databases of specimens from all over the world. I work in a museum, the Senckenberg Institute here in Germany, which means that we have large specimen collections of species of mollusks from all over the world, which allows me the opportunity when I have, say, a specimen of the Muar oyster in hand, I can go to my collection and I can line up all of the different oysters from all over the world and compare them side by side and see really easily, it's the same, it's different. No, this is really different. It's a new species. These resources are absolutely invaluable. And we have a major problem, which is that there is not a serious natural history museum in Malaysia. And yet Malaysia has all of the biodiversity. So there is a huge need for specimen collections and research collections in megadiverse tropical countries such as Malaysia, and not just one museum, but as many as possible so that all researchers can access these types of resources. And it means joining a global network of other museums that can share specimens, share insights, and share resources, as well as having reference collections that allow local researchers to do what I can do, which is to just walk down the corridor, pull out a bunch of oysters and compare them side by side to help me understand when a species is genuinely new to science. And also, I think it's important to note that there are very big, important scientific ideas that come from especially mollusk biodiversity and also specimen collections of mollusks. So mollusks actually are the fundamental data for some really important insights that we more or less take for granted. So one is the latitudinal diversity gradient, the basic fact that there are more species in the tropics than there are at higher latitude. The evidence for this is based on the distribution of mollusk species and other marine hard-shelled species. There are big theories like evolution through escalation, the idea that predator-prey interactions drive adaptations, drive species to develop new escape mechanisms, new defenses, and this can actually be one of the major drivers for speciation and for morphological change through evolution, and also the past history of life on Earth and the fact that there are mass extinctions. We always hear about the meteorite that wiped out the dinosaurs, but actually the original evidence for the idea of past mass extinctions comes from the fossil record of mollusks and hard-shelled, shallow water marine coastal animals, not the dinosaurs. The dinosaurs just came along for the ride. And all of these are based on data that's gathered from museum collections, having data from all over the world where you can have large-scale, big data comparisons that allow us to actually draw out these patterns. So this brings me to an important question that I get asked a lot, which is that if mollusks can do all these amazing things, they can adapt to all of these amazing body plans and these strange forms, can't they just adapt to climate change? Surely, if they're so diverse and they're so amazing, they're going to be fine, right? But this is a dangerous, misleading thing about mollusks, because the evolution that I talk about takes tens of millions of years. I mentioned this, that many of the species that we examine in Malaysia, these are lineages that were established maybe 50 million years ago. This is a very, very long time when the continents were literally in different places. To form a new species takes a minimum of 3 million years, on average about 10 million years. And climate change is happening on a completely different scale. Climate change is happening within our lifetimes on a scale of decades. This is 10 to the 6. It's 6 orders of magnitude different in pace. This means that coastal species, we cannot just expect them to take care of themselves because of their diversity and their own resilience. We need to protect them now. We need to recognize what we have and make sure that ecosystems, like the beautiful seagrass ecosystems that Prof. Muta was showing us, they need some kind of conservation to enable them to have the best chance to bounce back from disturbance. So, I wanted to give you some quick insights into Malaysian mollusks and why I love them so much. The Malaysian Malacca fauna is extremely species rich. It's very special, it's very diverse. And without taxonomy, without the hard work of actively looking at the identity of individual species, we stand a terrible risk of losing species before they're discovered. There are so many species that have not yet been named, not yet been discovered. And so we cannot develop strategies to protect them and to protect our natural resources unless we know what is already there. And also, as Prof. Muta mentioned, there is resilience in diversity. Ecosystems that are made up of more species are more resilient to change. And so it's important to recognize what species really are there so that we can understand how these ecosystems will respond to disturbance. So everybody can help. It doesn't necessarily mean that you need to be an expert taxonomist or an expert mollusk taxonomist, certainly, to help with understanding the biodiversity around you. Anybody can record what you see. There are now many online platforms to do really high quality biological record keeping. My favorite is iNaturalist. Anybody can join. Anybody don't even have to be a scientist. You can upload photos. Whether you are able to identify the species or not, it still helps to keep records of what species we see. It's important to recognize the value of these museum research collections and to support them. The new UPM marine collection and other museum collections that are starting at UMT and other institutions. And I would say to everyone, if you are working on coastal species in Malaysia, never, ever be afraid to question species ID. If you think something looks a little strange or something looks a little wrong or a little bit different or maybe not the same as it looks in the book, you're probably right. So please keep that in mind every time you're in the water. And I look forward to being in the water in Malaysia as soon as I'm allowed to travel. Thank you very much for having me.
Thank you so much, Julia. All right, so now we open for questions. It's amazing talk. I always love your talk. So, and of course, there's a lot of things that I'm familiar with because we are working together a lot. So, okay, one question. I already have one question from Dr. Nurusawa. She also works on mollusks. So her question is that can you advise us on the young mycologists, how do we work on taxonomy given the shrinking funds in this area, especially in Malaysia? And how can we keep the motivation and do this alongside with other branches of studies such as genetics?
Well, I think so. So yes, I know it can be very frustrating because it feels like you can't get started on doing serious work until you know what the species are, and yet it's almost impossible to get funding to figure out what the species are. And this is a very difficult problem to explain to government agencies and to funding agencies. And like anytime I can help with that, please call me. I'm happy to come in as an outsider to like try to explain this. Sometimes it helps. But you mentioned genetics, and I think one good way that is used all over the world is that we have the same problem with funding for fundamental taxonomy, and one thing that makes it easier to explain the value of taxonomy and the importance of taxonomy is to integrate it with many other different methods. So if genetics is what's possible in in your lab or in your institution, it's what's being supported, then use that to help understand the new species. If it's microscopy, then use the fancy microscope tools to try to understand new species. If you have the questions and if you have the organism in mind, then all lines of evidence are good ways to study new species. All of it is helpful and all of it can help unpick the puzzle of when species are the same or different.
Yeah. All right. Thank you. And I think one thing is very important is like sometimes you need to go to the right person. So mollusks is a very, very diverse group, and we cannot assume that everyone who study mollusks can memorize all the species. So sometimes what I do, just sharing the experience, is that I will always go to Julia because she knows so many people in the field, and she will guide me to the group of experts who are already working in that group. So by doing that, it's easier to narrow down, like especially for species identification.
This is a really good point, and there are many there's many scholarly societies. So there's there's Unitas Malacologica, which is the World Society for mollusk researchers. There's the Malacological Society of Australasia. These are really important networks to join, as member organizations, which help contact other experts, who know the local area or who know different groups of mollusks, and who, if they're nice, will put you in touch with other people as well. So, and I know that right now we're not really having conferences, we're not really having travel, but there's an email list for malacologists all over the world, and this is true for other groups as well. So most other big taxonomic groups, polychaetes, seaweeds, have email listservs with hundreds of people from all over the world who are often willing to to engage in collaboration or discussions. It's not okay to just like send a stranger a photo and be like, what's that? Because it's usually very difficult to answer that question, and that you don't know if it's the right expertise or if it's interesting for them. It's like asking somebody to do something for nothing. But if you say, I'm working on this group, can you point me to resources that can help me learn to understand it better, or put it in context of how exciting it is to study the biodiversity of a place as interesting as Malaysia, your particular ecosystem, your particular study site. That's a way to engage new collaborators who can bring additional expertise and help to understand the specific groups. Especially now, I would say, from a European perspective. So people think about Southeast Asia, and they think it's really hard to work there. Europeans look at Southeast Asia and they go, ugh, that's hard, because they've heard horror stories about how difficult it is to get permits to work in Indonesia, or how difficult it is to get permits to work anywhere, or how difficult it is to export specimens, from an Asian country to Europe, to do genetic analyses or to do morphological analyses here. What they don't think about is how valuable it is to actually collaborate with local people who also have expertise. So and this is what I always say to my European colleagues, I'm like, well, it's not a problem if you actually have collaborators in Malaysia who are actually helping with the research. Like, you guys have great labs. There's no reason to export very many specimens out of Malaysia, because you can do everything that needs to be done. You can do the genetic analysis, you can do the morphological analysis. Like, your lab is as good as my lab. There's no reason to move the specimens around. You just need the intellectual connection, to be able to share ideas and share expertise. And you can, reach out to collaborators from around the world and offer that, because actually, you have amazing resources, not just in your labs, but in the biodiversity and access to these incredible species.
Okay, thank you very much for that. So I think I can see that some of the audience are very happy and with what you just said, especially that you recognize that we do actually have that ability to do what we always have doubts in. So it's something that is very important. So I can see that there are some taxonomies in the audience. So I think it's not just for mollusks. They actually come from all over, and they study different group of animals. So is there any questions from the audience? If you have, please ask. You can always just ask. You don't have to type it out. But I have one question regarding the identification, because there's also a lot of people who are not familiar with the process in identification. They are not taxonomies, and even for me, it took some time for me to actually know how to do that. So if we found something, let's say we want to start, so we want to not only like ask someone, we want to understand the process. How do we actually identify something that we have in our hand? So other than just refer to books, other than just looking at in the publication, what else can we do? And what is the process actually looks like?
So this is hard. And it's, I would say the problem is that you almost need to be an expert before you can begin, so it is genuinely difficult. But in any difficult process, the way to manage it is to break it down into pieces that are a little bit easier. So if I go to the shore and I'm looking at something green, and I'm like, well, it's green. So my first question should be, is it seaweed or is it seagrass? But first, I just know that it's green, and green means plants. And so if I'm absolutely desperate, then I go and find, say, Prof. Muta, or I find, really any marine biologist, be like, what is this? And as soon as you know it's a seagrass or it's a seaweed, then you've already started the identification process. That's already an achievement. And the further that you can get, so usually we can do a little bit better than that. Most of us can do a little bit better than that. But once you've started the identification process, then you rapidly get to a point where you might have an identity that's useful. So you get it to family level, or you get it to genus level. And sometimes that's good enough. It may be good enough for whatever study you happen to be doing. And it also, when you get it to that lower level, you get it to just a family, or you get it to genus level, then you can access more detailed technical literature or look in a book. And then, that's where sort of the more difficult identification process starts. So what I would say is, don't panic about, like, nobody can just pick something up off the shore and immediately know what species it is. You can do this with your own local fauna for common species, but it's not the way that it works. You don't pick something up and just, nobody has this catalog in their head of 100,000 species. That's not a thing. So first you have to break it down into, like some broad group, and then a slightly smaller group, slightly smaller group, slightly smaller group. And it's okay to stop at any point.
Yes, exactly. Especially in Malaysia, sometimes we just do not have enough information to identify it to the species. All right, so we have another question from UMT, Dr. Nurul Afifa. Nice to see you again. The species name in worms and mollusk base sometimes have changes. And what is your opinion with the research that still have old names and but no longer accepted? So I think her question is like, why is there keep having changes in the worms?
Yeah, so I alluded to this in my talk, that this is a particular problem for Southeast Asian species. And so a lot of these changes happen because of ideas being updated. So when people go back to, taxonomists go back to study a particular group, and they study them in more detail, and it's realized that the material, the species that live in Southeast Asia are not actually the same as the ones that live in Europe. But unfortunately, because of the technical way that the taxonomy works, usually the European species get to keep their names and all of the Asian species have to change. I'm sorry, there's nothing we can do about that. It's the rules. It's not fair, but it's the rules. And so because of this historical legacy, the species names, yeah, they change a lot. And so this makes it a little bit difficult to trace sometimes if you're looking at historical literature and you're looking at modern accounts. It's sometimes not clear whether people are actually talking about the same species, right? So worms is a wonderful tool in that way because it will keep track. It doesn't just show you the accepted names, but it also will show you all of the previous names. And so you can check if it's the same genus and species name combination. You can look that up and it will immediately tell you it's not that anymore. Now it's this. So I try to stay with the current accepted name as much as possible. But whenever you have doubts about whether things really are the same name before and after, that's in a way that's actually the most useful thing that worms provides for us now, is these lists of synonyms.
Yeah, I agree with you. So the list of synonyms is very, very useful, especially when we look through the literature and compare to the current names. Okay, is there any question from the audience? I think we still have time for one more question. No? I think it's a very, very clear presentation. So, all right, Prof. Julia, I think I would like to ask you to give us like advice for us in Malaysia before you go offline, maybe before we move on. So what should we do? Because our taxonomy is really rare in the world, and it's even more difficult to train a young taxonomist in Malaysia. So what advice do you have for us?
So my advice has three parts. One is love your species. So the first is, I think that this is really important, in my work in Malaysia and in Southeast Asia, in any country, we never appreciate what we have at home, And so I can say this as an outsider, that in Malaysia, you have the most extraordinary biodiversity in the world, bar none. And so it's important for you to love and celebrate your own species and your own biodiversity. This is the most important thing. And also to inspire young taxonomists with this love and this enthusiasm for the extraordinary biodiversity that you have, because that's the only way that it will get worked on. It's the only way that it will get protected, is if you love it and cherish it. And then the second piece of advice that I would reiterate is that, you have extraordinary resources, both in terms of access to this amazing biodiversity, and also you have the resources, the infrastructure to be able to study it. I know there's this huge challenge to be able to cope with it because the diversity is overwhelming, but you do have the tools that you have in hand. And so do not be afraid to reach out to international collaborators, when you need support and when you need help, because it's not just, going asking for help. You have something to offer. You are offering really a bilateral collaboration that, other partners like me and many other people around the world should be happy to, be invited to.
Okay. All right. Thank you so much. On behalf of all the audience, thank you, Prof. Julia. All right, now we move on to the next presenter, the next speaker. Dr. Suchana Chavanich, are you here? Yes.
Yes, I'm here. Can you hear me?
Yes, glad to see you here.
Yeah, nice
All
to
right.
see you too.
All right, so before that, just an introduction to everyone. Dr. Suchana is an associate professor at the Department of Marine Science, Faculty of Science, Chulalongkorn University in Thailand. Her research focuses on the conservation and restoration of marine ecosystem, particularly on coral reef. And she and her team is considered to be the first group that successfully developed technique of coral mass culture using sexual propagation for coral restoration in Thailand. So she is currently the project leader of coral reef conservation in the Western Pacific under the UNESCO Subcommission for the Western Pacific. And as a scuba diving instructor and with the love of nature, she hopes to make a difference in protecting the marine ecosystem. So now we welcome Dr. Suchana with her presentation with her talk title, Coral Reef Conservation and Restoration Under the Changing World. Right, over to you, Doctor.
Thank you very much, Leena. Can you please share my slide, please? Okay, thank you. So, first of all, I would like to thank organizing committee for giving me an opportunity to join with you all today. So today I would like to share with you about what really happened in the coral reef ecosystem, particularly in the Southeast Asia regions. Next slide, please. And then I will share with you about our work related to the coral restoration in Thailand. And these are my co-authors who have been working, we have been working together for more than 10 years. Next slide, please. So, as we all know, coral reef ecosystem has the highest diversity compared to other marine ecosystem, and they are home to thousands of marine organisms. Next slide, please. So coral reef actually provide a number of valuable and vital ecosystem service that benefit people directly. Local people depend on the reef for fishery, shoreline protection, tourist business, livelihood, and more. But unfortunately, at present, as most of us know, that there are a local stressor. For example, rising the temperatures, seawater temperatures, rising the CO2, that making ocean more acid, and consequently corals are damaged, bleached, and eventually die. Next slide, please. So actually this picture shows us the local threats to the coral reef around the world. We can see that in Southeast Asia, we have quite red dot, right? Which means that we have quite a high threat in the area. Next slide, please. Yeah, when we zoom it closely, you can see that many areas in our regions are in the red color, red dot. So it means that our region's coral reef ecosystem quite in danger. Next slide, please. So this slide shows what kind of threat to the coral reef that we have in Southeast Asia. The example, all of us know overfishing, pollutions, coastal developments, and actually the degrees of the threats depend on the area. Next slide. Thank you. Wow. What do you think about this picture? Actually, this picture, I took it by myself. I can tell you in PP Island, I think many of you been there. In that time, actually, this picture, you can see just only 2 boats, right? And one boat have approximately about 200 or up to 300 tourists in each boat. But well, actually in that time that I took the picture, I didn't see just only 2 boats. I saw a total of 12 boats around me. Can you imagine that? 12 boats around me, and in each boat had about 200 tourists on the boat. And in that time, the reason I was there because I want to survey for the fish communities, looking, you know, how the distribution diversity. But actually, I end up seeing many feed. You know, many people feed just instead of fish. So well, one thing that we can learn is here that, you know, tourists can make and generate a lot of income to the country. However, if the management not good, if we not really have, you know, a limit of the capacity for the tourists, then coral reef can have some problem later on. Next slide, please. Marine debris is also a concern issue in the coral reef, as you can see here. And actually, marine debris not only have an impact to the coral, outside of the coral, but right now, actually, my research group and also other research team also found that many coral have actually microplastic inside of, you know, their bodies. So we're not sure what kind of effect would have from this microplastic inside of their body, but it would be very interesting to study more on that. Next slide, please. So unfortunately, recently, because of the global warming, right, that makes the water temperatures are rising. So we have frequently coral bleaching phenomenon, and I think 2 years ago, 2019, that we have quite a mass of coral bleachings also in the regions. But luckily this year we not. Next slide, please. And this hot water doesn't make only a coral bleach, but also other organisms, like in this case, sea anemone also got bleached. Next slide, please. And well, giant clam also get effects from this hot water or seawater temperatures rise. Next slide, please. So like other organisms, coral also, you know, they try to survive. You can see this picture. Most part are already dead, but still small part is alive. So they try to recover by themselves, but usually it will take quite a long time for them to be able to recover after, you know, for example, the coral bleaching phenomenon and other phenomena. So similar, I would like to give you an example, and I will give many people example. Is that similar when you are sick, you know, if you take a rest, okay, but if you not take a medicine, then you can still recover, but you know, slowly. But if you take a medicine, if there's something, you know, that can help you to stimulate you, then you will recover faster. So similar, like a coral case, which I will explain to you later on. Next slide, please. So scientists have predicted that in the next 30 years, if we not do anything, more than 90% of coral reef around the world will be at risk. It's like on the right-hand side of our screens. So, well, I think we all probably don't want to see the coral reef that we see now end up into the right picture that we saw now. Next slide, please. So as I mentioned earlier, there are several factors that contribute to the declining of the coral reef right now, but can they be able to recover back by themselves without helping from us, from human? Well, we are not sure. The reason why I say like that, because in the in the theory, they can recover by themselves, right? But unfortunately, right now, we have so many threats, so many factors contribute to the declining of the coral. And that's why I say that we're not really sure now that they can recover by themselves. And that's why right now, people or humans have to intervene, try to help to bring the coral back. Next slide, please. So what can we do? So actually, well, we can do either direct and indirect way. The thing that we and people always mention is that, yeah, definitely we have to have a good management, right? In order to bring the coral reef back from, declining or from the damage. So good implementation, try to reduce all those threats and then restore and rehabilitate the coral reef. I did say that the restoration and rehabilitation can bring those coral back as the same as before, but at least if we can help a bit, rather than let them recover by themselves, we can make sure that they can really recover back. It will not take for a long time. Next slide, please. Well, as we can see in our regions, coral restoration is not a new thing. We have been doing it for a long time, for more than 20 years. But however, right now, how we're thinking about restoration is a little bit chipping. Because of what? Because we know that, well, because of the climate change, and we know that at the end of the century, scientists predict that at least, the water temperature will increase about 5 centigrade. So now, many scientists who work on the restorations are a little bit concerned that, well, if we're doing a restoration, but then we not really consider the climate change, we not really consider the water temperature will rise, what would happen if we just, culture them and then we're not gonna know, or we really not gonna make sure that our baby that we coral can be able to survive in those, changing the temperatures environments. Next slide, please. As I just mentioned to you earlier, when we're talking about rehabilitation, restoration, first thing that people think is a marine protected area, right? Yeah, marine protected area is very good strategies for coral conservations. But you can see here, well, it's very interesting. Look at almost the end of the line, Southeast Asia, our regions. We have very high number of the marine protected area. Well, 599 marine protected area site, okay? But if we look closely, whether those 599 sites have effective management or not, well, I think some of you are probably not going to surprise. We have just only 2% of those 599 sites that really we can call effective management or effective marine protected area. So what this means is that, yeah, marine protected area is good strategies, but a lot of time, because of the budget, because of the manpower and those things, that makes our regions a little bit hard to make those MPA effectiveness. Okay, next slide, please. Okay, next slide, please. As I just mentioned at the beginning, that what I also would like to share. Next slide, please. Oh, I think you skipped one slide. Can you go back? Okay. Yep, that one. No, forward one, please. Sorry. Okay, so in terms of active restoration techniques for the coral restorations, in Thailand, actually several techniques have been used to restore coral reef, but most of the time they use a sexual reproduction technique, breaking the coral into the small pieces and reattach on a new substrate. So actually this slide showing the the difference between sexual reproduction and asexual reproduction. I think some of you already know. So sexual reproduction, how we do is that, because the coral spawn egg and sperm, so we collect egg and sperm and then fertilize, artificial fertilization, and then raise them into the certain age, and then we release them back. Okay, but for asexual reproduction, is opposite. Is that we don't care about egg and sperm. What we do is that we break the coral into pieces in order to grow them and let them form a new colony. Next slide, please. This is the example of the coral restoration that can be found in Thailand and also in Southeast Asia. Okay, we can transplant the coral in a whole colony, or do a nursery, floating cage, make them from a small fragment, and then when it go into a certain size, not yet. Okay, when they go into a certain size, then we can transplant them back. Okay, or I know that in Indonesia, but in Malaysia, I don't think you have this one yet. Using electric currents, try to stimulate the coral, the juvenile coral, to settle on the substrate. Okay, but we use a low electric current, but not strong one. If strong one, coral would not survive, and the diver would not survive too. Okay, so we have to use the low electric current. Next slide, please. So there's also questions that, well, can the coral cover be restored in the absence of nature recruitment and reef recovery? Well, the answer is yes. They can, even though they in that area don't really have a nature recruitment. But important thing is that, well, the environment there have to be good, good conditions for the coral to grow. If we transplant, if we try to restore or rehabilitate the coral reef, but in that area, the environment is still in bad conditions, well, there's no point to do this kind of work. Next slide, please. Okay, well, I think several techniques of coral restorations, many of us probably have heard for quite a while, quite a long time. But the things I would like to share with you is recently it has been quite popular in the regions, in our regions. So by using the this technique, sexual reproduction technique, and as I mentioned to you, we collect egg and sperm from the field, and then artificial, fertilize it not in the lab, but actually if you want to make a mass culture, you have to do it in the hatchery. So this is an example that we do it in Thailand. So we first succeed in fertilization in 2006, so about 10 something years ago. But even though now we still develop, try to develop the technique in order to meet with the circumstance, which I will share you later on too. So now we can culture more than 10 coral species through this technique. Next slide, please. So how we do is that we collect egg and sperm when they spawn at night time, okay? And usually, as we all know, they spawn just once a year. So we try to, make sure that we can collect enough in order to be able to fertilize that, and then we bring it back to our hatchery. Right now, we have 2 big hatchery in the Upper Gulf of Thailand with the collaboration with the Department of Marine and Coastal Resources Thailand. And after we artificial fertilize it, we raise them in the hatchery for 2 years. Have to be for 2 years, because from our research, we found that if we release them, less than 2 years old, then they would not survive. But if we raise until they're about 2 years old, then they can be able to survive quite significantly. Okay, next slide, please. So many people probably have some question. Well, for 2 years, how old is the baby? How big of the baby coral will be? So actually, usually our corals grow about 1 centimeter per year. So 2 years is about 2 centimeter. Well, even though it's not too big, but is big enough in order to release them and allow them to survive in the environments in the Upper Gulf of Thailand. Next slide, please. Well, and what happened? Actually, now I'm not just only have kids, I'm become a grandma now, okay? Because the baby coral that we produce, well, after 5 years, they mature and they can reproduce again. So this is quite a success. Successful things that we are pleased because we have been working for a long time, more than 10 years, and eventually we found that, well, after 5 years they can be able to reproduce again. Next slide, please. So now we can see that it's a closed circuit. So we can start produce, from egg and sperm, and then raise them, and then transplant them, and then they can be able to, those parent colony can be able to reproduce again. Next slide. This slide just want to show you how about other country in the regions. Japan also doing this kind of technique. Philippines are doing this kind of technique. But you can see that Thailand take about 5 years to become mature. Philippines just only 3 years. I'm not sure why. Maybe they do some stimulation, mature faster. For Japan, 4 years. But for Thailand, it's probably a little bit late, 5 years, but it's okay, but if they still can be able to produce, that's fine. Next slide, please. So since we are in the changing world now, having to face with the climate change, as I mentioned earlier, then we have to be a little bit careful now that, and want to make sure too that the baby that we produce or the culture that we try to propagate in a mass, propagation need to be adapted to the climate and thermal stress too. Next slide, please. You probably heard about supercoral in the past couple of years. People talking about this supercoral, yeah. So what it means is that, well, how we can make the coral to be a green man, like a Hulk, is very important, right? If we culture the coral, we have a baby, and then eventually they die because, the water temperature, that is gonna raise over time. So now people think a little bit, one more step ahead is that we have to produce the baby coral like a Hulk baby. Next slide. And actually, well, good news is that Thailand, now we can do it by training them, and this teaches that actually coral, people think that, well, coral, you can train them. Yeah, they can train them. Originally, many people would thought that zooxanthellae would play a big role for, bleached or not bleached coral. But in the reality right now, yes, zooxanthellae clade can play a role, but the adaptation by coral themselves can play a major role in terms of whether they can withstand to the, risings of the water temperatures or not. Next slide, please. The other things that in just only a past couple of years, it has been quite important issue, and people try to work on it, cryopreservation. I'm not sure whether many of you heard about it or not. Cryopreservation is a technique for freezing the tissue or cell to preserve for use at a later date. Okay, so cells are stored at ultra low temperatures. Okay, and this technique actually is quite important now because what we learn, because of the climate change, higher sea water temperatures, many coral now suffering is that they cannot really reproduce as it should be. So if we add this cryopreservation for conservation, that will help the coral in the future, just in case if they cannot spawn anymore. Next slide, please. So cryopreservation, you know, we do a lot in human, in vertebrate, but coral just only in the past couple of years that we try to do it. Next slide, please. And well, good news is that not just only in Thailand, not just only my group, but other group also, we can preserve the sperm using a cryopreservation. Now we try to work on eggs. So if we can, in case, hopefully in the next couple of years, also if we can also preserve the egg too, that would be perfect. So it means that we can bring the egg and sperm from the cryopreservation anytime and try to do artificial fertilization in case if those coral species, you know, have kind of endangered. Next slide, please. And well, this have been issued since 2 years ago that we succeed in, you know, our cryopreservations for the coral sperm. Next slide, please. Okay, so what is the future for cryopreservation in terms of coral conservation, restoration? Now, people try to more focus on the coral cryopreservation techniques because these also can add to have more effectiveness for the coral conservation and restoration in the future under the changing world, under the climate change. Okay. So last slide, please. So remember, now when we talk about coral restoration, coral conservation, and coral rehabilitations, well, we have to make sure that we, okay, not only, you know, organizing the wedding ceremony for the coral, yeah, not like a wedding organizer, but we want to make sure that we are a family planner, that the baby should come as a Hawk baby. Okay, thank you very much. Thank you.
Thank you so much, Dr. Suchana, for your interesting talk. So this is the first time I heard about cryopreservation using in coral. So I think it's an amazing technology that maybe it can help a little bit, especially when the corals have problem with climate change that they cannot survive. Maybe you will be able to produce a more resilient coral later. All right,
hopefully.
So now, yes. Right, so now we move on to questions. So we already have a question from Dr. Nurul Salwa. So she mentioned that she asked that what are the best medium used in coral transplantation, either PVC, cement, or any other type of materials?
Well, very good questions. Well, the substrate, as you see, but it really depends. In the past, people use a lot of PVC, right? Because it's cheap, it's easy, you know. But start from a couple of years ago, because of the microplastic, you know, it's an issue. So right now, in terms of coral restorations, we try not to use the PVC. If we can prefer concrete is better for as a substrate, okay? But that is mean that you have to pay quite a lot of money for that. So actually, variety of substrate can be used, but have to make sure that those substrate not gonna cause any additional pollution to the ocean.
Okay, so is there any study on the breakdown of PVC used for the coral restoration? Is there any breakdown analysis on whether the microplastic actually come from the PVC itself?
Yep.
Okay.
I know what I heard. Yep, right.
Yeah.
And they concern it now.
Okay, that is a very bad news then.
Yep, right.
Okay, so Dr. Nursawa has another question. How do you manage the corallivorous animals such as the Drupella snail or COT, crown of thorns, from eating the corals?
Oh, well, so very good question. Fortunately, in the upper Gulf of Thailand, we don't really have a Drupella. We don't have much. So we not really, when we transplant those baby coral, we're not going to have this issue, Drupella or, you know, caught a carnotron. But the problem that we have actually is fouling organism. Why? We have to make sure that the baby coral that we release, back have to be, grow enough to be able to compete with flowering organisms. You were surprised in Japan when they culture the coral, they release them just only 2 months, or 2 months, not 2 years, 2 months.
Wow.
And because Okinawa don't really have a lot of flowering organism.
Okay.
So in Southeast Asia, we have a problem with the flowering organism. So that's why we have to take a longer time in order to to release them.
Yeah, I agree that in Southeast Asia we have much richer water with more organism in the water.
Yeah.
All right, so we have another question from Samantha Lisa Durit. So she said, interesting presentation, Prof. What is the most endangered coral reef in the world or in Asia? I think which one is the most endangered coral reef in the world or in Asia? Her question.
Oh, you mean the area?
Yeah, I think area, coral reef.
Wow, very good questions. Well, from I talk with other scientists, actually, in the past, many people worry about all regions, right?
Mm-hmm.
Because coral have been decline, because of all anthropogenic activity. But if you follow the news in the past 2 years, the one that have most effects will be Australian reef. But I don't want to pinpoint that. But now we try to, discuss what really happened is that because of coral can adapt themselves, right? So sometimes, but this have to be proved, okay? Sometimes if the reef is too much protected, well, the results might reverse. You understand what I'm saying? Yeah. So right over region, unfortunately, see, there's only 2% effective MPA. So they really have to survive by themselves. But these have to be, looking through again. But we surprised that in the past 2 years, Australian reef have in dangers. But I would say in danger, but effects from those, coral bleaching and so on. But right now, we not really know much about really what is going to be in dangers.
Okay. All right. So we have another question from Dr. Abby Wu. So I think have Dr. Suchana seen any possible strain of coral that are particularly resilient?
Good question. Oh, question is very good. Very difficult question.
Yes, Dr. Abby Wu's question.
Yeah, right. I know him very well.
Yes.
So, right now I know that many scientists also trying to find the strain of the coral that is, kind of a tough strain. But as I mentioned earlier, when we're talking about strain, the genetics, now people working on it. But if not genetics, adaptation can play also an important role. It doesn't have to be strain. It's how the coral adapt themselves can make those coral resilient to the environments. Okay.
Yeah, I agree. From what I see from my own experience, because I live just beside the Strait of Malacca. So Malacca Strait is very, very busy strait with a lot of shipping every day. So the sedimentation in this area also is very, very bad. So, but what we observe is there are some corals somehow survive in this environment, even though most of them are gone. We can see the decrease in the population, but there are some that are still thriving. So I think this coral may hold some secrets in how to be resilient. Maybe they have a special gene or special things that we can learn a little bit from them.
Yep.
Yeah. All right. So any more questions from the audience? So maybe not. All right. So we only have 2 more minutes. So 2 more minutes. I think maybe Dr. Suchana can give us closing remarks on your talk, like maybe some words of encouraging and advice for us.
Well, last but not least, I would like to thanks all of you for coming today, and I hope you enjoy my talk. And last, also, I would like to give to you is that coral reef is very important, okay? And as a scientist, you know, we cannot work alone. We need partners. And hope, you know, if you are one of, you know, a scientist that are working in these areas, please reach out to different stakeholder partner to make it, you know, effective as we plan.
Yeah, thank you very much. All right, I think I would like to thank all our speakers. So our speakers, all our speakers make a very, very important point. That is where we cannot work alone, especially in marine science, because as the sea is connected, there's no separation in between, even though no matter how narrow is that. So in order to achieve great work, we have to make sure that we work together. All right, thank you very much, Prof. Harah, Prof. Julia Sigwart, and Dr. Suchana. Thank you so much for sharing your experience. We are looking forward to the next webinar so that we can learn more from all these experts. And I think I want to call for a round of applause, but they have to do it virtually. All right.
Okay, thank you.
Thank you so much. All right, thank you everyone for joining us. If you want a e-certificate, don't forget to fill in the link that is shared in the chat box. Okay, Prof. Harah is here again. All right, thank you everyone.
Okay, bye-bye.
Bye.
Okay. So I can leave now, right?
Yes, thank you.
Before you leave, can we have a photograph?
Yeah,
Oh,
sure.
Yes, I hear all the speakers.
Yes. Oh, Dr. Julia left.
Oh, hi, Abby.
All right, please turn on your video if you would like to take a picture and be part of our picture group photo. Right. Okay, anyone else? Yes, some are slow. Right, I think more is coming. Okay. Okay, I think Dr. Firdaus, are you taking a photo? Okay, 1, 2.
Need next frame, please. Okay, but for the next frame, the people didn't. I mean, like our viewer didn't open their video, so
Yeah.
So I will just screenshot their name as a record.
Julia said she is coming. She will join
so
again
back
for the
to
picture.
the main
All right,
frame.
just give her a minute. Right, she will join us again for the photo. Yes, she is back.
Okay, wait. Yeah, smile, everyone.
Dah terlebih smile dah ni.
Thank you, Patutlah.
All right.
This is so sweet. Okay, now I'm done with the main frame with all live videos sharing. Now I move to the next frame where only name appeared here. Thank you very much, everyone.
You're welcome. Mak Raya. Raya yang seronok.
Seronok bro. Dua kat rumah.
Raya yang budget.
Interesting Raya, you know?
Okay, I'm done with all frame. Thank you very much, everyone. Thank you for your patience.
Bye. Thank you.
Angkat kain, hujan. Bye-bye.
Okay, thank you.
Bagus awak.
Okay, done.
Done. Finish. Okay, bye.
Selamat
See you
pagi,
next
Prof.
time.
Bye.
Thanks, Prof.
Julia, bye. Thank you very much.